Hi. Okay, so today's video is going to be about DNA replication. So we're going to title our notes DNA replication. And before we get into that, I would like to remind you that the reason why DNA replication needs to occur is because sometimes cells need to replicate. And so when a cell needs to replicate, it needs to first replicate all the DNA inside the nucleus. And so DNA replication occurs during the S phase of interphase of the cell cycle. So I'm going to say this process occurs in the S phase of interphase of the cell cycle. Okay. And so if you remember in the cell cycle, what will happen is the cell is in G1 first, and the cell spends most of its life in G1. That's a phase. And then when the cell decides it needs to replicate for either for growth or for injury, then it will go into the S phase. And in this phase, all the DNA replicates inside the nucleus. And then it goes into G2, and then it goes on into either mitosis or meiosis. And so this process occurs in the S phase. Okay, and so to explain this process, I'm going to explain it in 4 steps. And I'm going to explain it by introducing you to each enzyme that does each step. Okay, and so this is a process that involves lots of enzymes. And so I want you to picture in your mind that it's a chromosome, and that chromosome is going to turn into 2 chromosomes that are identical. Okay, and so what happens is the very first thing that needs to happen is I'm going to draw a picture of a DNA strand. Okay, and if you remember, this is a double helix. Okay, and so I'm going to draw it like this. It's a twisted ladder. And so step 1 is that this twisted ladder needs to be straightened out. And the enzyme that does that is called topoisomerase. So topoisomerase is an enzyme that straightens out the twisty helix. That straightens out the twists in the double helix. Okay, and so what that looks like then is then we have this, oops. Oh my goodness. Okay, so let me move that back here. That goes here. Okay, so then what we have is once it's straightened out, we're going to have both sides of the double helix in a straight line. Okay, so I'm going to draw the topoisomerase as teal, and it's an enzyme that is moving its way through the double helix, straightening it out. Okay, so this is the topoisomerase here. I'm going to put an arrow here that shows you that. Okay, so then now that the DNA is straight, then the next enzyme comes in, and that next enzyme is called helicase. And helicase then splits the 2 strands apart. So step number 2 is helicase is an enzyme that separates the 2 strands. Okay, because the double helix has 2 strands, one that is complementary to the other, and there is going to be an enzyme, which I'm going to make red, and that is going to split up the 2 strands. Okay, so I'm going to make this helicase here. Okay, and I want to point out that this is moving upwards. Okay, and this is also moving upwards. And so helicase is here. And so I'm going to point to the helicase here. And then now I'm going to show you that once the helicase has opened the 2 strands, it looks like this. Okay, so here's the left strand. Whoops, I don't want an arrow. Here's the left side. Okay, and now it has just one of the bases in the base pairs, and then here's the other side. Okay, so I have 1, 2, 3, 4. Okay, and so I want to point out also that each of these nucleotides has a top, which is the 5 prime, and a bottom, which is a 3 prime. And so I'm going to just write right here on the side. Well, I'm going to draw one nucleotide. Okay, this was in the last video that you watched. Here's the phosphate group. This is just one nucleotide, and then you have the deoxyribose sugar, and then that is attached to a base. I'm just going to make it an adenine. And so this top part is the 5 prime side, so it's the top, and the bottom is the 3 prime side, okay? And then when you add a new nucleotide, you always have to add it on the bottom, on the 3 prime, okay? It can only be added here. So the next nucleotide is going to be here. Oops. Okay, and it's going to be the phosphate group of the next nucleotide. So I'm just going to quickly draw this. Here's our sugar. And then this is another base, so let's just make it a T. Okay, I'm just picking random ones. And so this is the 3 prime end, okay? So there's a top and a bottom to each nucleotide, and there's a top and a bottom to the whole strand, okay? So on this side, I'm gonna make this up here the 5 prime, and this down here is the 3 prime. And in a double helix, you always have the opposite orientation going. So because this is 5 prime over here and this is 3 prime over here, on this side over here, you have a 3 prime, and then down here is the 5 prime. Because in DNA, they're complementary on both sides, but one is going in one orientation and the other one is going in the opposite orientation. Okay? And so this helicase is just going through and it's unzipping the DNA. It's opening them up. Okay, just like it's showing here. Okay, the next step is a very important enzyme. And this one is the most important enzyme of all. And this is called DNA polymerase. And DNA polymerase is the enzyme that builds new DNA. So I'm going to say that makes new DNA. So there's going to be this new enzyme that comes in, it's going to build new DNA based on the old DNA. So these two strands are split apart, and each of these strands, which are from the old copy, are going to be used as a template to make a new copy. Okay, so what I'm going to draw is that DNA polymerase is going to start. I'm going to make DNA polymerase hot pink, and it's going to start here. Okay, and it's going to move its way in this direction. Oh, I'm sorry. I got to erase that. I got to start over here. So the DNA polymerase can only build new DNA from a 5 prime to a 3 prime direction. Okay, I'm going to write that in the notes because this is an important point. Okay, DNA can only be built in a 5 prime to 3 prime direction. Okay, and that is because just like I drew over here, you have your nucleotide and the top is the 5 prime, and you can only add the next nucleotide on the 3 prime end. Okay, so the DNA is being built like this. You can't put a nucleotide on the top here on the 5 prime. So it's always being built from 5 prime to 3 prime in that direction. And so over here on this new DNA that we're building, on this side on the right, the new DNA has to go in this direction. So it has to be built in this direction. Whoops, I didn't mean to do that. So it has to go in this direction because it has to go from 5 prime to 3 prime. And because this is 5 prime up here and 3 prime down here, that means the new DNA has to be 5 prime to 3 prime in that direction. So the new strand, which I'm going to make blue, is going to be built like this. So here's the new DNA, okay? It's going to be built here, and then here, and then here, and then here, okay? In that direction. So let's say, let me put some letters on here. Let's say this is A, this is T, C, C, okay? What that means is that this right here has to be T, and this has to be A, and this has to be G, and this has to be G. And then when the new strand is being built, this right here is going to be A, this is T, this is C, this is C. Okay, and this is one new strand of DNA now. And this new strand of DNA has one half of it being new, built by DNA polymerase, and one half of it being old, which is the original parent strand. Okay, so there's an old side and a new side. And the same is going to be true on this side. And so because DNA can only be built 5' to 3', it has to go in this direction then. And so I'm going to put the new strand here, it's blue, and it has to go in this direction, where this side is the 5' and this side is the 3'. And so this C is going to go with G, this C is going to go with G, this T is going to go with A, and this A is going to go with T. And so now we have two strands that are identical. And you can see that over here on the left, you have an AT, and on the right, you have an AT. On the left, you have a TA. On the right, TA. On the left, CG, 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 CG. Okay, do you see that? Okay, so what I'd like to point out here is that this DNA polymerase, which I'm going to make hot pink, okay, it started here for the right side. And what it does is it moves in this direction, going up. And it's following the helicase. So as the helicase unzips the DNA polymerase is right behind it, building new DNA. This strand is called the leading strand. Okay, so I'm going to write this down. There's two strands. One is the leading strand, and this side is where DNA polymerase builds new DNA following the helicase. Okay, so the helicase is unzipping, the DNA polymerase is right behind it, building new DNA. The other side, however, is called the lagging strand. And the lagging strand, the DNA polymerase has to start here because it can only go 5' to 3', and then go in that direction. So opposite of helicase, it's going backwards. And so what's going to happen on this left side is this is going to do the opposite in the backwards direction, and it has to wait for the helicase to unzip some more. Then it's going to go up to there and come back down backwards. Okay, so that's called the lagging strand. And because the DNA polymerase has to do it in a backwards way, and it has to jump back up and do it backwards way, it takes longer on this side. So that's why it's called the lagging strand. So this side is where DNA polymerase builds new DNA in the opposite direction of helicase. I'm going to say in the backwards direction from helicase. Okay, so it's going opposite. And so that's called the lagging strand. And so it's going to make this little piece right here. So it's going to make this little piece, and then it's going to jump up here and make another little piece. Then it's going to jump up here and make another little piece. So the lagging strand is making its new DNA in pieces. Okay, so the new DNA is made in pieces, and these pieces are called Okazaki fragments. In pieces called Okazaki fragments. Okay, and this is named after a Japanese scientist who figured out how DNA replication works. And he would see all these little pieces of DNA, and he thought he was making a mistake in the lab because he kept finding pieces of DNA, and he's like, I keep cutting the DNA up. But it turns out that that's part of DNA replication. And so it's named after him, and they're called Okazaki fragments. Okay, so then step 4 is the last step. There is a fourth enzyme called ligase, and ligase is the glue. And what ligase does is then it glues together all those Okazaki fragments, okay, because they're not a continuous piece. There needs to be an enzyme that comes in and pieces them together. So ligase, the glue, puts together all the Okazaki fragments. Okay, and then so in the end, So in the end, you have two identical strands of DNA, each with a old side and a new side. Okay, so each one is half old, half new. Okay, I think that is everything. Okay, and because one side is old and one side is new, this is called semi-conservative. Okay, so it's half old, half new. So it's not all old, or that would be all the way conservative. It's just semi-conservative. Okay, so that's a term you have to know. Okay, so that's it for this video. I hope everyone's doing well, and I will see you next time.